So I'm going to be presenting this paper, What Animal Is This Anyway? Assessing Data Quality in Undergraduate Student Faunal Reports, on behalf of myself and my collaborators, Suzanne Needs-Howarth and Trevor J. Orchard. And this is part of an ongoing fun and productive collaboration, some of which you saw an example of in the previous paper by Trevor. So again, I'd like to begin with a land acknowledgement. Today, I'm joining you from the University of Toronto, Mississauga, and I wish to acknowledge this land on which the University of Toronto operates. For thousands of years, it's been the traditional land of the Huron-Wendat, the Seneca, and the Mississaugas of the Credit. Today, this meeting place is still the home to many Indigenous people from across Turtle Island, and we're grateful to have the opportunity to work on this land. We'd also like to acknowledge that the land on which this research was conducted encompasses the traditional territories of and contemporary homelands of many Indigenous people whose presence has shaped the history and land for thousands of years. I want to begin by asking why we should care about data quality in student reports, particularly for people who are non-zooarchaeologists. So let's take a step back for a second and think about this. And in Ontario, every year, we generate significant amounts of archaeological data. Much of this comes in the form of analyses that are included in license reports, because, as you know, the ministry has reporting requirements for Zooarch, for archaeobotany, and for other things as well. So all of these analyses are done. They go into the reports, but we can ask the question, how often are these data used for anything other than fulfilling ministry license requirements? Certainly, sometimes we do see that things move from gray literature to publication, but often this is in the form of site-level descriptions. So we see this data as an amazing potential resource for addressing big picture questions, such as was just described by Trevor in the previous paper. These can be questions about the relationship of people to the land, how that may have varied over time, questions about how people and other species adapted to new conditions, whether they be climatic or cultural, and so forth. So this is great, but before we start mining these old reports, we need to have confidence in the data quality in those reports, whether we're looking at things like flake counts or descriptions of lithic raw material or ceramic types or faunal identification. So although it might seem really kind of boring, we would argue that an exercise in a data quality assessment is needed before we begin those large meta-analyses. And so today, what I'm going to do is outline an example of this using student zooarchaeology reports. Okay, so many of you, I hope, will remember the well-loved and inspirational Dr. Savage. Dr. Savage taught a course that many Ontario archaeologists completed. It was a course in zooarchaeology. It was remembered for its rigor, for its breadth, and for the production that every student had to do of a faunal report. Dr. Savage taught this course for 22 years. And in that time, about 400 reports were produced. Many of these, possibly most of them, were from samples from Ontario. And the ideal was that every student would complete identification of 500 bones to a below class level for their final report. And then once they produced that final report, it was back in the day, okay, it was given to the archaeologist who provided the sample. It might have been used in archaeological reports, and a copy of it also went into this like bank of filing cabinets, which in Howard's time could be accessed by other researchers. So that bank of filing cabinets represents an enormous amount of student effort. if you just do basic math, we're looking at something like possibly as many as 200,000 identifications. Not all of them from Ontario, but a significant number that are just sitting there in student reports. Other people have noticed this. So many years ago, in fact, the OAS published a summary written by Janet Cooper and Howard Savage in 1994, just describing all of the sites and the kinds of animals that were present on those sites. And then, as Trevor mentioned earlier, the bird data was summarized in a publication by Douglas Sadler and Howard Savage posthumously. So as indicated, we're interested in looking at bigger picture questions, looking at things like regional fishing patterns and distribution of passenger pigeon over time. So we've been doing this so far using our own data and data from other established zooarchaeologists, such as Rosemary Previck, who very generously shared her data. But maybe we're a little bit greedy. We wanted to know, would it be possible to add the analyses from these hundreds of reports to some of our analyses? So we set up an experiment or a study to try to figure out whether this is going to be possible. So there's an old picture of the Faunal lab with one of those filing cabinets. And so what we did was we looked at 5 analyses from Dr. Savage's class, and we compared them to analyses that we'd done of faunal samples from the same sites. And the sample sizes vary a little bit. So the dotted bars here are the student analyses, and the large, the not dotted bars are the ones that we did. We have one case in which mostly the identical set of bones was analyzed by us and by the student. And then in the other cases, we have very large samples that were analyzed by us and smaller samples that were analyzed by the students, but they weren't the identical, they weren't from the identical larger sample, okay? And we think that the recovery methods for this one, site B, were identical between the two samples, but we know that for site C that the recovery methods were not the same. So that was what we wanted to do, is compare their findings with ours and see how they landed. Now you might ask, before we go any further, you know, like, who's to say our identifications are right and the student ones are wrong? And that would be a fair question. And so we do have data from another study that we've done where we've looked at known identity specimens. And we've asked experienced zooarchaeologists and students, essentially, or people with less experience, to identify these objects. And this is one that, this is a graph that shows identification rate over time. We gave them 50 things to identify, and the people who had more experience, so those are the ones with the little asterisks here, given enough time, were able to obtain a very close to correct identification rate, whereas the people who, with less experience, even with a lot of time, weren't doing quite so well. So that's why we think that probably with our experience, we have figured out how to identify bones. So our first question in this was, if we just look at class level data. So here we're just asking the most very basic thing. If we're just asking, how are their classifications of bird, fish, mammals, reptiles, and amphibians compared to our own? Are we getting the same patterns? So in the sample where we analyzed almost exactly the same thing, we see that they're very close. So that's great. In the second sample, where we had the same recovery methods but different samples, we see that our data and student 2 are pretty close, but student 1 is a bit different. Possibly this has to do with just sampling, because if we look at this, the difference lies in fish versus mammal. And we think that those would be difficult to confuse. Now, things get really ugly when we look at Site C, where we see that the students are both identified a much higher proportion of mammals compared to our identifications of fish, which incidentally look very much like Site B. We think that this probably arises from recovery methods where our data was recovered on window screen and would have had a very high recovery of fish that might have been lost in larger screen mesh sizes. Okay, so at the class level, not too bad. We wanted to see next what kind of data would students be able to extract from faunal samples. So once you've got your pile of material that's fish, how many of those, or what proportion of those bones are you going to be able to identify as something more specific than just fish? So can you say it's a salmonid or a catastomid or something like that? So here what we found is that in our data, we had a lower proportion of bones that were left at only the class level. So we were able to identify more, or a higher proportion, to something more specific than fish or mammal. So that's true for Site A, and that's true for Site B, and it's not true for Site C. And again, that might have to do with recovery methods. So from this, it's worth thinking about what the reasons for this would be. And we're going to suggest that possibly if the analyst is inexperienced and may have a poor grasp of the animals that would be available and or the skeletal anatomy of the animals, then it's possible that they're going to systematically exclude or be unable to identify some taxa. And so their lists of identifications won't be as representative of what is in the sample than the list of somebody who's more experienced. So we would expect a lower degree of diversity in the student analyzed samples. Okay, now we're going to look at the things that they were actually able to identify below class. So for those of you who aren't zooarchs or who aren't archaeobotanists, this legend at the top goes from most general to most specific. And so really what we're interested in here is how precise were the data. So if you are not a faunal analyst, it's important to know that it's not possible in many cases to obtain an extremely precise taxonomic identification because there are similarities in the skeletons of related data, related taxa rather. So for example, when an analyst says that a bone belongs to a smallmouth bass, it is possible, depending on the bone, that it could also come from a largemouth bass, but maybe the analyst didn't check. And so by calling it a smallmouth bass, rather than just saying it's a bass, they're inserting more or asserting more precision than's actually possible. So analysts who have a lot of familiarity with the range of animals in an area and also their skeletons will often identify at this higher level of order or family and not identify at the genus or species. So they're more conservative. So if we look at this graph, what we see is that in the student data, often they have a higher proportion of identifications to the species level or to the genus level. And the our data has a greater proportion of identifications to the order or family. So we're being more conservative. We can see this, for example, with the sucker family, where this isn't true for all of the data sets, but for some of them. In our data, in all three, we've got a large proportion that are identified only to family. Whereas in site B, you have all of them identified to genus, and in site C, either genus or species. And this isn't true just of fish. So we also saw that there were overly precise, overly precise identifications of other taxa as well. So within mammals, we saw this is true for mice and voles, and also between dog and wolf. And we saw this in birds. Ducks are notorious for being difficult to distinguish, and in some cases they were identified at a very specific level. And we saw this within fish. Very closely related species, sauger and walleye, were distinguished. And then we also saw it, interestingly, in terms of the actual elements. So in some cases, there was a very specific identification of elements that we would argue are not diagnostic enough to be identified. So, so one thing we noticed is that there was a tendency of the students to be more precise in their identifications than we would argue is justified. Now, what was the impact of this on actually a couple of these things, on the overall picture that the students were generating about the types of animals at these sites? So we're going to look at fish because, as you probably figured out by now, fish interest us. And so there are four fish families that are frequently found in significant number in Ontario. And we see that for the most part, the student data and our data recognize the presence of all of these four fish families. I will note that student 2 at site C completely did not identify any of the bass or sunfish in their identifications. We also noticed some more subtle differences. So for example, suckers are more important in our data set than in the student data sets. And the other thing to point out is that for all of the student data, these fish families make up almost all of the identifications of fish. Whereas for us, they're slightly lower. And so this is explained in the next slide, where we look at other notable families. So it's important to note that the scale is changed here, okay? So we're looking at bowfin, eel, pike, and salmonid family. And we see that in our data, these make up a variable, but notable proportion of the identifications, whereas in the student data, they're either absent or almost absent. This likely arises from the systematic exclusion of fish vertebra from identifications, which was the trend in the lab back in the day. So what it means is that if we're looking at those student reports, we need to be aware of the fact that animals likely to be represented only by their vertebrae are probably not going to be found in the summaries. So we would have to be very careful about any overall distribution of fish family data. Okay, misidentifications. So in addition to overly precise identification and non-identification, we also found some examples of completely wrong identifications. So for example, identification of a long-tailed field mouse which is a European species and would not plausibly be found on a site in Ontario. But happily, these kinds of misidentifications were rare in the student data, and they're also fairly easy to spot and exclude, because if you've got experience, you'll realize, oh, what's this thing doing here? So that wasn't a big issue. So to return to our question about whether or not we can use student-generated data for larger projects, I would say not as such. So we can't simply walk in and start taking out those reports from the bank of filing cabinets and using the data in them. But there are, we have been able to put together a list of things that are indicators for problematic data. And so we can use this list, and there's a couple others that I didn't include, to evaluate the reports. And I think this is useful not just for student reports, but also for other kinds of reports. So we've got faunal reports that are going into ministry license reports, and if we don't have any information about how the analysis was done and who did the analysis, then examining the data set for this kind of indicators can help us get a sense as to whether or not these are data that we want to use in larger meta-analyses. Furthermore, I think doing this kind of exercise for people who might be interested in questions related to lithic raw material use or something like that, doing an exercise like this would allow those people to use their data for, or lithic data or ceramic data for meta-analyses as well. So we shouldn't assume that all the reports are going to be the same or equally good or equally bad. Writing of those reports was a learning process, and possibly the identifications made early in that process aren't as reliable as identifications made late in the process. And I think that probably not all of the data, as Trevor alluded to earlier with the passenger pigeon paper, not all of the data is equally problematic. So it's just a question of being careful when you go about using legacy data sets. Okay, so I want to thank Max Friesen for providing access to several of the faunal reports. This was actually done during COVID, so he had to go in and get them when everything was closed down. We'd like to thank Max for access to the faunal lab, and we acknowledge and are grateful for the privilege of studying belongings from the ancestors of the Huron-Wendat and other Indigenous communities in Ontario. Hopefully I didn't go too long.